Hi, hello, namaste. Welcome to Shrav's Biotech for You. Today we'll discuss about the types of cloning vectors. The first type is plasmid. Plasmid is nothing but a small, circular, double-stranded DNA lacking protein coat. It is 1000 to 2 lakh base pairs in size. Higher the copy number of the plasmid, higher is the number of plasmids per host cell. And of course, more the copies, more is the protein that will be synthesized. The first plasmid vector that we will discuss in this class is PBR322. Coming to the nomenclature of PBR322. P stands for plasmid, BR stands for Bolivar and Rodriguez, who were the two scientists or the researchers who developed it. And 322 is nothing but the order of the synthesis of this plasmid, and this distinguishes from other plasmids that were developed in the same lab. This is the diagrammatic representation of PBR322. It is 4.4 kb in size and it has one ori, that is the fragment of plasmid PMB1. This acts as the origin of DNA replication, and it has two selectable markers, that is two antibiotic resistance genes, ampicillin resistance gene and tetracycline resistance gene. Coming to the cloning sites, it has number of unique restriction sites like PST1, EcoR1, HIND3, SAL1, etc. that are present within the antibiotic resistance genes. Cloning into one of these sites inactivates the gene and allows the differentiation of recombinants from the non-recombinants. This insertional inactivation is being discussed in the previous class. Coming to the pedigree of this PBR322, nothing but the origin of each part of this cloning vector. Ori, as I said, is the fragment of plasmid PMB1. This PMB1 is closely related to the colicin producing plasmid called E1. Coming to the selectable markers, ampicillin resistance gene, it is originally present in the R1 plasmid. R1 plasmid is the naturally occurring antibiotic resistance plasmid in E. coli. Tetracycline resistance gene is derived from R65. This is the second antibiotic resistant plasmid. Then the cloning sites. As I said, there are many unique restriction sites within the antibiotic resistance genes in this PBR322 cloning vector. Now moving on to the advantages and disadvantages of PBR322 vector. First advantage is small size. It is approximately 4.4 kb in size, hence it facilitates easy purification and manipulation. It has two selectable markers, hence it facilitates easy selection of recombinants. It can be amplified to 1000 to 3000 copies per cell by stopping or blocking the protein synthesis using chloramphenicol. Coming to the disadvantages. The first disadvantage is high motility. The vector may get lost in the population of mixed host cells. The second disadvantage is the limitation in the size of the gene of interest that can be inserted or that can be accommodated. It does not have very high copy number, and the screening process is time consuming and laborious. With this, I end today's session. We will discuss the second example of plasmid vector in the next session. Please like, share and subscribe to my channel.